Hi everyone, welcome to video 2 of the medulloblastoma meta-analysis portal. If you want more information on the portal, check out the first video on summary plots that I uploaded earlier, and additional links are provided in the description. In this video, we will look at how to perform Cox regression, visualize matrix, methylome t-SNE visualization, lollipop visualization, sample view with disco plots and methylation array data with the genomic locus view. So let's start since this video has a lot to get into. So the first one that we're going to do is Cox regression. To do Cox regression, you go to the charts tab and click on the regression analysis button. For this tutorial, we are going to run Cox regression. And this is a statistical technique used primarily in survival analysis. What it does is it assesses the effect of several variables on the time it takes for a specified event to occur, such as the event could be death or failure or any other event of interest. So once we click on the Cox regression analysis button, the first option that pops up is for selecting the outcome variable. Since we have survival analysis as the measurable outcome, that is the variable that we are going to select. We will select progression-free survival. Now, there is the term information provided for this term. If you click on it, you can go to it and see it. Now, progression-free survival is a term used in oncology to describe the length of time during and after the treatment that a patient lives with the disease, such as cancer, without it getting worse. Now, we choose this to get a real assessment of how well a treatment is working in the clinic. And so select that term. And then the next option it shows you is to select the independent variable. And well, you can also see the number of patients within the progression-free survival term. So there are 664 patients for whom the disease has not progressed or it has not become worse. But then there are 234 patients out of these 898 samples for whom the disease has progressed. So now let's select an independent variable. And this is a variable that might affect the progression of the disease. So it gives us more information about the overall progression of this disease for those particular patient groups. So here I can go ahead and select some variables from the dictionary tree. So let's select demographics such as biological age, and then another term for age group. And we can also select clinical variables. So I'm going to go ahead and select molecular group here. Now, what we want to do is make sure that our reference is the category within these variables, which contains the highest number of samples. So I have selected those as the reference, basically. Now, why we do that is because that is the category that we are controlling for. And the other categories within that group, that variable is going to be compared to that reference. So the reference is going to basically act as your control here. Now, the other thing I also want to do is include univariate analysis. Now, this helps examine the impact of single predictors for this outcome. So make sure to select that and then click Run Analysis. So what you get is this table, and this will show up with the results. And you can see here that for each variable, there are separate rows. So there is one row for biological sex, and the category within that also has the number of events and the number of patients in there. So there's 307 female samples in here for biological sex with events around 62, right? And on the far right, you can see there is univariate versus multivariate analysis. So how to interpret this data now? So we can see within molecular group. Okay, so let's look at one row at a time. So we can see within molecular group, the hazard ratio for sonic hedgehog and group 3 is the highest within that group. Right now, this is being compared to the reference, which is group 4. So what this is telling me is that for these patients within sonic hedgehog and group 3, they fare much poorer in terms of the progression of the disease as compared to group 4. And the demographics, on the other hand, such as the biological sex and age group, don't seem to have much of a significant impact on the progression of the disease. And this is displayed by the hazard ratio, the lower hazard ratios. And the confidence intervals on these are also pretty narrow, which also gives me more confidence on the significance of these metrics. So that is shown by the p-values. So for Sonic Hedgehog, you can see it is slightly significant, that's 0.02. For group 3, it is even more significant. So Sonic Hedgehog and group 3 could account as single predictors for the progression of the disease for this analysis. So within Cox regression, in the next video, we will also look at how to use custom variables to perform some complex regression. Now let's move into another visualization, which is your sample matrix. Now, sample matrix, as the name suggests, is a matrix or oncomatrix view of the mutation, copy number, data, as well as dictionary variables. To see what that looks like, click on this sample matrix button that is shown here and load the pre-built matrix. So to load the pre-built matrix, you have to click on the matrix plot button. And now you can see that this is getting updated. There are about 117 rows, I believe, which is what that is loading. And now you can see this row by column matrix where each row is a variable. And these variables could be clinical, demographic, or genomic. And each column is a single patient sample. So in this colorful pre-built matrix, I am going to zoom out here a little bit. And I can also zoom out from the matrix plot. You can see the labels are on the left side of the matrix. So you have different panels for different things. So on the top, you have all your clinical and demographic variables. And then you've got cytogenetics below that. Then copy number variation, all mutations. All mutations would combine your somatic and germline. And then you have a separate panel to visualize the germline mutations as well. Now, these labels themselves also have clickable events. And so you can click on each of those labels and configure, right? So you can click on any label. Here I'm clicking on OTX2, and I can either edit this, replace this, or remove this, or I can launch the lollipop plot from here, and we will see what the lollipop plot is. Now, what I'm going to do is pick the top gene here, which is beta catenin, okay, CTNNB1, that codes for beta catenin. And I'm going to click on that, and I'm going to sort all my samples against that gene by using this left arrow. I am going to remove the sequencing row from here because after I sorted that, it shifts down. Basically, that is showing me the data availability for sequencing for these samples. So I have just removed that entirely. And now you can see that this beta catenin is showing somatic mutations or missense mutations for 88 samples in here. And this is the most prevalent mutated gene in the Wnt group. And once I have sorted for this, I can go back up to the cytogenetic panel and see that there is a monosomy for chromosome 6. So there's a loss of one copy of the chromosome 6. And this is a classic feature of the Wnt molecular group. So there are additional settings on top of the matrix view which a user can use to customize. And for example, you want to customize the view of the genomic alterations or to customize current gene set, right? So how do we do that? So you can go up to these certain settings that are present right on top of this matrix. And let me click on the genes and let me show you how you can edit your gene set. Say you want to, remove or add additional genes. So what I can do is from this drop down, I can select all mutations and I can click on edit select group, and this displays this UI panel to me with all the genes that are currently loaded onto this matrix. So I can choose to click on these genes and delete them, or I can add my own genes. So let me add KRAS here and hit submit. Now what I see is the matrix reloads again, and I can see KRAS somewhere in here. Now it does not have any mutations, which is why the row looks empty. But you can, in this way, you can add genes or variables if you want to. And I will remove that. Next, I want to segregate this cohort into sub-cohorts to be able to compare the individual groups within and stratify these samples further. So to do so, I'm going to go up to the control settings and then click on samples, and then I'm going to group samples by molecular group. And again, the chart is reloading, and I can now truly confirm whether the monosomy 6 that I was seeing for chromosome 6 is a feature of Wnt or not. And you can see that this sits right under this category of the Wnt group for medulloblastoma. And so you can, in this way, you can customize this matrix chart and be able to see all the genomic, clinical, and demographics of a patient and stratify it further to run specific analysis of interest. Now let's look into how to visualize and use the methylome t-SNE plot, right? I will leave this open because we are going to come back to visualizing the lollipop through the matrix. But let's look at the t-SNE. Now, this portal has methylation data for 898 patients, using which the classifications for these molecular groups and molecular subgroups have been performed. Now, there are t-SNE classifiers used for this sort of visualization. And on top of that, unsupervised machine learning algorithms have been used to classify these patients into one subgroup versus another. Now, to understand more about this disease context and these classifications, keep an eye out for the final video where I will actually get into the biological use case. But classifier information and other methodologies can be found in the paper links that are provided in the description below. So let's look at what this methylome t-SNE is all about. Now, this methylome t-SNE is housed under the scatter plot. So if you click on the scatter plot button under the charts tab and click on this first option for methylome t-SNE, it will load this pre-built t-SNE that segregates these patient samples shown by dots here into the 4 different molecular groups. So blue is your Wnt, red is sonic hedgehog, group 3 is shown by yellow, and group 4 is the green dots. Now you can see that group 3 and group 4 boundaries are not very well demarcated, and this is because of the complex heterogeneity of the disease, and we will also get into that in the final video. But to further stratify these patients spatially, we can use this burger menu where all the settings for this scatter plot are present. Now, what I'm going to do is under the color, I'm going to replace that to stratify this based on the molecular subgroup, and I will go ahead and hide that control panel. Now I am seeing these 12 different subgroups present within those main 4 groups. And what I want to do is I only want to focus on the group 3 and group 4. So I can choose to hide some of these subgroups through the legend. And how I can do that is by clicking on each of the legend items and choose to hide them. So I'm going to hide Wind. I'm going to hide Sonic Hedgehog as well. So now I can zoom in using my trackpad, and I can see the individual dots for group 3 and group 4. And if you did notice, there are 3 samples that have been clustered together with the Wnt subgroup. And that's because of the classifier. They are potentially misclassified, but we don't know that. So let's zoom in and look at these dots for group 3 and group 4. And what I can see is that each of these are different molecular subgroups, right? So you have, if you hover on these dots, you have group 3 and group 4, subgroup 1, and then subgroup 2, subgroup 3, subgroup 5, subgroup 6, subgroup 4 is somewhere here, subgroup 7 are the light blue dots, and then green are the subgroup 8. So 8 subgroups are present on this methylome t-SNE. And hovering over each of these dots will give you information about the sample and the molecular subgroup. Now, if you click on any of the individual dots, it also gives you more clickable options to view this sample in the sample view, disco plot, or methylation array. So let's click at sample view to look at what this sample is about. Now, now going from a subcohort level to a sample level data will give you all the information about that individual sample in this portal. And you can see the survival outcome for this patient, that this patient was alive after 11.6 years of uh, after getting enrolled in this study. And we can also see that this patient is aged around 6 years old. They fall into the category 3 to 7 years, and they are a female. And you can also look at other information about them, which is more clinical features, cytogenetics, whatever you might want to look at. On the right is a disco plot or a circos plot, which visualizes the copy number data for on a chromosomal level for this patient. So copy number gain is represented by the red, and copy number loss is represented by blue. So you can see here they don't have any mutation, but they do seem to have certain copy number events. And these copy number events can be visualized on the methylation array at a probe level data, where this methylation array is showing an entire chromosomal genomic view where each of these dots is a single probe. And so this is sample level data, and within this sample level data, there are different types of visualizations that you can pick and choose based on what you want to visualize and represent. Now, on the methylation array, we can see that on chromosome 17, and let me zoom in here a little bit so it's more obvious to see. You can see that on chromosome 17, there is a gain and also a loss. So this is technically an isochromosome 17. So 17p loss versus 17q gain. And if I click on that, this will take me to the locus view on the genome, right? Now this is a position chromosome 17, and I can zoom out to see the entire chromosome and see where this copy number loss or gain is. And I can see along the axis of this chromosome with my reference gene indicated by green, there is copy number gain as well as copy number loss. So this is a case of isochromosome 17 shown by these vertical bars. I can confirm that this is an iso 17 chromosome. And also, the guardian of the genome, TP53, is present on chromosome 17. So I can use this search box additionally to look at TP53 and see whether there's any loss or gain events on over the gene locus. So I can zoom out, and for this patient, what I can see is, so TP53 is in a reverse order orientation, and around the promoter region, I am seeing a gain and some loss as well around that. So these are the kind of things that you can look at for through a methylome t-SNE and go into details to visualize single sample level data for individual patients. Now, the next thing I can also do is group samples together to perform a more rigorous analysis. Now, how do we do that? So what I am going to do is select this lasso tool. Now this tool has been provided to make selections on top of the t-SNE. So I'm going to go ahead and circle around subgroup 2. Once I do that, this shows me certain options. I can choose to list the samples. I can choose to add them to a group. I can choose to add them to a group and a filter, or I can show those samples within sample view. So I'm going to go and add these to a group, and you can see the number on my groups tab changes from 0 to 1 because we just added these samples to a group. Now I am going to select my next group, which is subgroup 7. And here as well, I am going to select add to group. So now I have two groups in here, right? My first group was subgroup 2. So I am going to go ahead and rename that under the groups tab. And then I am going to rename my other group as subgroup 7 because that's what we selected. And this is used for creating custom variables. Now, you could add more conditions by using these and or buttons, and this will open up the dictionary tree again, where you can select more variables to add in more filters to narrow down your subcohort even further. Or you can directly go, which I am going to do now, is create variable using two groups. So I have created this custom variables shown by this blue label. And what that would do is I can use that for further analysis. Now this shows up under the charts tab. If you click on dictionary, you can see this subgroup 2 versus subgroup 7 as a custom variable, right? And if you click on it, it will give you a summary of the distribution of that data in a bar chart. And what I showed you previously in the video for creating these summary plots and overlaying terms, you can easily do that with custom variables as well. But let's get back to this groups tab where we have this custom variable button, because this is where I'm going to run some other analysis, right? So if I click on that label, I can see the number of samples within each of those subgroups. And now here's where I can do further visualizations. I can choose to display a matrix plot. I can choose to compare survival between the two groups. I can choose to summarize it, or I could choose to open the methylome t-SNE to show these samples on the scatter plot, or I could delete this variable and start all over again. So let's look at the survival analysis for this. So I'm going to click on compare survival and then progression-free survival. So we chose progression-free survival in Cox regression, and that is what I'm going to do here. So here you see a Kaplan-Meier curves for a progression-free survival with the probability of survival plotted against the time to event, which is shown in years. And I am going to go ahead and edit some of these x-axis ticks. So I'm going to add 0, 5, and 10 here. And then I'm going to shut this control panel. So what I have here is subgroup 2 shown in green and subgroup 7 shown in orange. And as you can see, it is quite significant that the survival outcome of subgroup 2 versus 7, the subgroup 2 seems to be doing much poorer or much worse compared to subgroup 7, right? And after adding these 0, 5, and 10, I can hover over each of these time points to see the difference between the survival outcome for these two subgroups at 5 years of the time point. So what you can see here is at 5 years, subgroup 7 has around nearly a 90% survival without progression of the disease compared to the 65-66% survival in subgroup 2. And this gap narrows as we move towards the 10-year survival time point. And as the time passes, you can see that there is a slight drop in the survival outcome for subgroup 7, whereas the curves are relatively stable for subgroup 2. And below this x-axis, you can see the legend for subgroup 2 versus subgroup 7, which shows the number of patients outside of the parentheses, and in the parentheses are the ones that are censored. Now, what are censored patients? Censoring occurs when patients leave the study or they're lost to follow up, or the event of interest, which is progression in our case, has not occurred by the end of the study period. So what you see here are the number of patients outside the parentheses, and then the censored data is shown in the parentheses. So we can see that at each time point from 0, 5, and 10, the number of patients are increasing in the censored group. And it is increasing a lot more for subgroup 7 versus subgroup 2. And that indicates that, for example, at 5 years, there are 57 patients for subgroup 2, and 10 of those were lost to follow up, they were censored. And then for subgroup 7, there are 124 patients, and 15 are lost to follow up. And so in this way, you can visualize survival analysis, which shows you these Kaplan-Meier curves, and you can choose to make some changes in the settings and then use that for your visualization. So now we can move into visualizing variant level information for individual samples. And for that, we can use the lollipop plot. The lollipop plot can be accessed in several ways, right? The main way to access that is through the genome browser under the charts tab. But what we can also do is we can use the matrix that we had loaded previously to visualize the lollipop plot. So I'm going to go here and select one of the genes. And then if I click on the lollipop plot, that should open a lollipop plot for me, showing me variant level data for different samples. Now let's look at how to use the genome browser button. What I'm going to do here is type in CTN NNB1 for beta-catenin, and then I am going to load this genome browser view for beta-catenin. And we can see the lollipop chart right in front of us. This lollipop represents the individual variants with the number of cases highlighted for those variants in white numbers within the lollipops. The horizontal rectangular box is the length of the protein, with different vertical rectangular regions shown in these colors. Those are the different domains that are present on this protein, and those are also shown here in the legend. And this legend is interactive. You can choose to hide or show a domain by clicking on it, or you could also add your own protein domain if it is not represented in here. So now note that all these mutations are somatic. All of these mutations are shown by these lollipops, and these are also clickable. And these mutations are somatic in origin, and all of them are missense mutations shown by the blue. And what this is telling me is that these are mutations that are all activating in nature, thereby hampering with the ability of beta-catenin in one way or another to regulate Wnt signaling. And to see the exact nucleotides and genomic position of these mutations, we can drag across this region on this lollipop plot, and that will allow me to zoom into this hotspot region. And here I can see that this is exon 3 where these mutations are, and amino acids 30 to 41 are what are implicated for these mutations. And you can also see the individual codons for each of these mutations. Now let's look at the individual sample level data within these variants. And to do that, click on any of the lollipops, and that will open this sunburst with the info button. So I'm going to go and click on that info button. And once you click on that, you should be able to see a table with rows for individual patient information. And this information is distributed across molecular, clinical, and demographic levels. So you can see that for this sample, it belongs to the molecular group and subgroup Wnt. You can see the age, you can see the biological sex, and the variant that they have, and the origin of that variant. What it also shows you is the genomic position and the change in nucleotide that occurred at that positions. Now, if I click anywhere else on the chart, that table disappears. And now I can also choose to stratify this visualization based on filtering criteria. Now, say I want to filter and look at males and females separately. So I can do that by clicking on this sample button, and then I can go to biological sex and I can click on female, and I can do the same step sequentially for visualizing males. Now, these are two separate sub-tracks that have opened below the main tracks. One is for female and the other one is for male. So now what I can see from here is that certain variants are restricted in a certain category. So I can see that this D32H is only present in female, there's only one sample, but it is not present in males. But whether this is real or this could be an artifact of a low sample size. But in either case, you can use this lollipop chart to draw certain inferences and stratify patients based on variant level information. And that's all I have for this video. In the next and final video, we will investigate that cross communication within the portal using an actual biological use case that I found for my first author publication during my role here at St. Jude's. We will additionally see how to create custom variables by combining clinical, demographic, and genomic variables to stratify patient, run some more complex analysis, and how to set up filters to create subcohorts. So keep an eye out for the final video in this medulloblastoma meta-analysis portal series.